In order to understand the nervous system, we must first understand the basics of nervous tissue. The nervous system is our master controlling and communicating system. The cells are going to communicate via electrical and chemical signals. These signals are rapid and specific and provide an immediate response to stimuli. The general functions of the nervous system include monitoring changes both inside and outside of the body. This is our sensory input. We will need receptors that will pick up the various stimuli that tell us of changes both inside and outside the body. This sensory input will go to integration centers. The integration center will process and interpret this gathered information and make decisions about what we need to do. Output will go along the motor system. This motor output will provide a response. It will activate some sort of effector organ. These are going to be muscles or glands. Here is an example. Your thirst receptors tell you that your mouth is dry. You see a glass of water. That would be sensory input. The integration centers put this together and say, we're thirsty, we see a glass of water, let's pick up the glass of water and drink. So motor output would cause you to be able to pick up the glass of water and take a drink from it. The nervous system can be organized structurally. We have the central nervous system, or the CNS. This is composed of the brain and the spinal cord. The peripheral nervous system, or PNS, is composed of the cranial nerves, those nerves that come directly off the brain, and the spinal nerves, the nerves that come directly off the spinal cord. The nervous system can also be organized based on how it functions, and this really only applies to the peripheral nervous system. The central nervous system's only function is that of integration. But the peripheral nervous system has to take care of both input and output information. The sensory division is the afferent division. This brings in the sensory information to the central nervous system. This is the input part of the system. We can have either somatic sensory nerves that pick up external stimuli. This tends to go to our conscious brain and we're aware of the stimuli. Visceral sensory input takes care of internal stimuli. Typically, we are unaware of this, so this goes to our unconscious brain. The other branch of the peripheral nervous system is the motor or efferent division. This is our output division. This sends information to the effectors. The somatic nervous system is driven by our conscious brain. This goes to the skeletal muscle. The autonomic nervous system is driven by our unconscious brain. This takes care of events that we don't think about on a daily basis. The autonomic nervous system is divided into the sympathetic nervous system. This takes care of emergency types of situations, and the parasympathetic nervous system. This takes care of day-to-day activities. So as we look at our divisions, we have the central nervous system, brain and spinal cord. Its function is integration. The peripheral nervous system is where we see functional division. We still have the cranial and spinal nerves as the structures, but we have the sensory division, which takes care of input. That input is sent to the central nervous system for integration. The central nervous system sends output to the peripheral nervous system, which goes to the effectors. We have both somatic fibers, which pick up external stimuli, and the visceral fibers that pick up internal stimuli. On the motor side, the output side, the somatic nervous system takes care of voluntary activities. This would be our skeletal muscle. But the autonomic nervous system takes care of our internal unconscious kinds of activities. Peristalsis, heart rate, breathing, things like that. The autonomic nervous system is divided further into the sympathetic system. This is going to mobilize body systems in emergency settings, or the parasympathetic nervous system, sometimes referred to as our housekeeping system. Nervous tissue is composed of two principal cell types. We have the neuroglia. These cells support, protect, and insulate the neurons, which is the second cell type. The neurons are the nerve cells. These are the ones that conduct nerve impulses, so we say they are excitable and conductive. There are 6 types of neuroglia. 4 of them are found in the central nervous system: the astrocytes, microglia, ependymal cells, and oligodendrocytes. 2 are found in the peripheral nervous system: the Schwann cells and the satellite cells. The astrocytes are the most abundant glial cells in the central nervous system. These cells help support and anchor neurons to their blood supply. They're also responsible for the blood-brain barrier. They very carefully control what substances leave the blood and get to the nervous tissue, so they're highly protective. The microglia are small, highly branched cells that are mobile. They move around the central nervous system and act as phagocytes. They respond to damage and clean up damaged tissues. This is sort of the internal immune system of the central nervous system. The ependymal cells are found lining cavities of the brain and the spinal cord. Most of them contain cilia. They assist in circulating the cerebral spinal fluid. They're also involved in maintaining something called the blood-CSF barrier, yet another barrier that helps control what has access to the nervous tissue. And the oligodendrocytes are the ones that form the myelin sheath in the central nervous system. One cell is capable of wrapping as many as 60 neurons. In the peripheral nervous system, we have the satellite cells. The satellite cells surround the cell body of the neuron. The Schwann cells are the ones that are responsible for forming the myelin sheath in the peripheral nervous system. It takes several Schwann cells to wrap one axon. The neurons are the structural units of the nervous system. They conduct nerve impulses from one place to another, and they also live an incredibly long time. If you take care of them, they last your entire lifetime, which is good because they lose their ability to divide, so we can't replace them if they get damaged. Neurons have a very high metabolic rate. They have a great need for oxygen and glucose, and they must have glucose. They can't fall back on other forms of nutrition. There are three basic parts to a neuron. The dendrites are processes that extend away from the cell body. The axon is another process that extends away from the cell body. The neuron cell body, sometimes referred to as the soma, is the area of the cell that contains the nucleus, the nucleolus, a lot of the cytoplasm, and many organelles. This is the biosynthetic center of the neuron. All of the proteins, bits of membrane that need to be replaced, and chemicals are synthesized in the cell body. There is a lot of rough endoplasmic reticulum found in the cell body. At first glance, this looked like a totally different cell structure, and it was given the name Nissl bodies. But as we got better microscopes and could see this structure better, we realized it was just rough endoplasmic reticulum. While not its major function, the cell body can be a receptive region of the cell. Most neuron cell bodies are found in the central nervous system. The neuron processes are things that extend from the cell body. We have two types of processes, the dendrites and the axon. The dendrites may be hundreds of short, tapering, diffusely branched processes. They will have the same organelles as we see in the cell body. They'll have cytoplasm as well. Their primary function is to receive signals. They're capable of being irritated at their dendritic spines. They will conduct these impulses to the cell body using something called graded potentials. While there are many dendrites per cell, there is only one axon per neuron. The axon emerges from the cell body at a thicker area of the cell body known as the axon hillock. The axon is what accounts for the length of the neuron. Axons will branch profusely at the end or the terminus. There can be 10,000 terminal branches at the end of an axon. These distal endings are called axon terminals, synaptic bulbs, or terminal boutons. Impulses are conducted along the axon using action potentials. They will secrete neurotransmitter at the axon terminals and use this to signal the next cell. So as we look at a neuron, we see the cell body with the cytoplasm, the organelles, the large nucleus, numerous short, highly branched dendrites that receive impulses. Here is the axon hillock and the very long axon that will branch at the end to the axon terminals. The axons will contain all of the organelles except the Golgi and the Nissl bodies. This means that the axons cannot manufacture or package proteins. They have to transport these from the cell body. They use special cytoskeletal elements to do the transporting. Anterograde transport is when materials move away from the cell body toward the axon terminals. This is how neurotransmitter, which is manufactured in the cell body, will get to the axon terminals. Retrograde transport is when we move toward the cell body. As neurotransmitters are broken down in the synapse, pieces are sometimes reabsorbed. These materials use retrograde transport to get back to the cell body so they can be remanufactured as neurotransmitters. The central nervous system is very well protected. It's very hard for materials to get into the nervous tissue. However, certain viruses and bacterial toxins can damage neural tissues using this retrograde transport to reach the cell body. Polio, rabies, and the herpes simplex virus, as well as the tetanus toxin, can enter at the axon and use retrograde transport to get to the cell body. There they can damage the neuron. There is research being conducted to use retrograde transport to possibly treat genetic diseases. If we can engineer a virus that contains the corrected genes or some sort of microRNA that can suppress a defective gene, we can use this to enter the central nervous system and therefore correct the problem. The myelin sheath is simply an insulator. It's composed of fat and it helps separate the signals from neighboring axons. Just like you don't let bare wires be part of an electrical circuit, we don't let bare axons be part of an electrical circuit. Myelination will increase the transmission speed of the impulse. If we say a nerve is myelinated, they have myelin sheaths. Axons are myelinated and have very fast impulse transmission. Unmyelinated fibers do not have a myelin sheath. Dendrites and cell bodies are unmyelinated, so they have very slow impulse transmission. There are a few small axons in the central nervous system that are also unmyelinated. In the peripheral nervous system, the myelin sheath is produced by the Schwann cells. It takes several Schwann cells to wrap one axon. The cells do not overlap each other, so there are spaces between the cells that are known as the nodes of Ranvier. As the cell wraps around the axon, the cytoplasm and organelles are pushed to the furthest point, so only the outer layer of the myelin sheath contains the cytoplasm and the organelles. Here you see the axon, and all of these little marshmallow puffs are individual Schwann cells, and you see the points where there's no overlap. These are the nodes of Ranvier. The cell will begin wrapping itself around the axon, and as it does, we will get cell membrane, cell membrane, cell membrane. Only the outermost layer will have the cytoplasm and the organelles. Think about having a plastic bag, a Ziploc bag that's got just a little bit of water in it. Start wrapping it around a pencil. You will see that the water squishes away from the pencil, and for a while you're only wrapping plastic bag parts around it. It's only that very outer layer where you get some water in that wrapping. That's how the Schwann cell works. In the central nervous system, it's the oligodendrocytes that are responsible for the myelin sheath. They send out multiple flat processes, not the whole cell, just some of their cell membrane. One oligodendrocyte can wrap as many as 60 axons. This means that there is no outer collar of perinuclear cytoplasm or organelles. There are also more widely spaced gaps in the myelin sheath in the central nervous system. When we talk about white matter, these are simply myelinated fibers, axons. When we have a collection of axons in the central nervous system, we call them tracts. In the peripheral nervous system, they're called nerves. Gray matter is unmyelinated tissue. This would be your cell bodies and your dendrites. When we have a cluster of gray matter in the central nervous system, we refer to that as nuclei. In the peripheral nervous system, that cluster of gray matter is known as ganglia. Here you see the oligodendrocyte sending out processes to wrap multiple axons. So we still have that fatty sheath, cell membrane, but there's no cytoplasm or organelles in the outer layer. Neurons can be classified both structurally and functionally. When we look at the structural classification of neurons, it's based on the number of processes. A multipolar neuron has one axon and several dendrites. This is the most common and major neuron type found in the central nervous system. Bipolar neurons still have only one axon, but they also have only one dendrite. These are relatively rare. Some of our sense organs have bipolar neurons, things like the retina or the olfactory mucosa. Unipolar neurons are probably not truly unipolar, so they talk about them being pseudo-unipolar. They appear to have one process that comes off the cell body, but it branches to an axon and a dendrite. It appears that the axon and dendrite are fused, but they're probably not truly fused. Here we see some of the different types of neurons. The multipolar with several dendrites but only one axon, the bipolar with one dendrite, one axon, and that unipolar. It looks like the axon and dendrite are fused. They probably really aren't, but we still have one axon and one dendrite. The functional classification of neurons is based on the direction in which nerve impulses travel with relationship to the central nervous system. There are three types: sensory or afferent neurons, motor or efferent neurons, and interneurons. Sensory neurons are input neurons. They pick up information from sensory receptors and take that information toward the central nervous system. Their cell bodies are found in the ganglia of the peripheral nervous system. Ganglia are simply clusters of gray matter, or cell bodies in this case. Most are unipolar, and they're also referred to as first order or primary neurons because they are the primary input for the information to the central nervous system. Motor neurons or efferent neurons are output neurons. They carry information information from the central nervous system to the effectors. Their cell bodies remain inside the central nervous system in the somatic system. In the autonomic nervous system, it's a little more complicated. Mostly, these are multipolar neurons. Interneurons, sometimes known as association neurons, are connectors between sensory and motor neurons inside the central nervous system. They're how some of that integration information gets around. The entire neuron of an interneuron is found inside the central nervous system. About 99% of the neurons in the body are interneurons, and these are primarily multipolar. The sensory or afferent pathway is getting information into the central nervous system, and we have both visceral and somatic incoming information. If a visceral neuron picks up the stimulus, this is going to go to our unconscious brain. The stimulus is detected by a visceral receptor, something inside one of our internal organs. That first sensory neuron, that first order sensory neuron, will carry the information from the receptor to the central nervous system. This neuron is in the peripheral nervous system. Once in the central nervous system, we will have a second order neuron that will transmit this to the part of the brain known as the thalamus. The thalamus is sort of the switching center for everything. Initially, it gives everything a good or bad feeling, and then it decides whether it goes further up the chain or stays within the unconscious brain. With a visceral neuron, the signal stays within the unconscious brain. If it's a somatic neuron that's activated, we're going to know about this. This is going to go to our conscious brain. Again, the stimulus is detected by a sensory receptor, and a first-order neuron, something from the peripheral nervous system, transmits the stimulus to the central nervous system. Here, a second-order neuron will pick it up and take it to the thalamus. We'll still get that good or bad feeling thing, but the thalamus will decide this time to connect it to a third-order neuron in the central nervous system. This will take the stimulus to our conscious brain, where we will interpret it as touch or sight or sound. The motor or efferent pathway is when our conscious brain signals for movement or our unconscious brain signals for some sort of smooth muscle activity, heart muscle activity, or glandular activity. This starts with an upper motor neuron. This is inside the brain, and it will transmit the stimulus to a lower motor neuron. The lower motor neuron will leave the central nervous system. The lower motor neuron from the central nervous system to the peripheral nervous system takes the message to the muscle or the gland that needs to be activated. That activated muscle or gland then responds to the stimulus.